So yeah, a very warm welcome also from my side. And again, thank you for EBIP inviting us to this tech exchange talk today. My name is Stefanie Kiderlen, and today I will talk about multiphoton microscopy in life science. I would like to start to give a brief overview about our company. So Prospective Instruments was founded in 2019. We are a young company located in Dornbirn, Austria, which is very close to the Swiss and Austrian border, what you can see on the map here. And the company was started with the idea of building or providing turnkey, flexible, modular, very compact and also affordable multiphoton microscopes. We have 4 different divisions on our company. So the first division is Prospective Life Science, where we have the multiphoton microscopes. We have a fully integrated version, which is called the MPX, I will talk about today. And then we have the MPC, so a customized version for everyone who already, for example, have their own lasers. We offer the scanning optics only. The second division is Prospective Lasers, where we have femtosecond lasers. The lasers we build in the microscope. We also offer a standalone devices for different applications like multiphoton imaging, optogenetics, film, cars, and so on. Then we have a third division, it's the imaging service. So we offer contract imaging service with different microscopes, not only multiphoton, but also film microscopy, whole slide imaging, light sheet microscopy. And there we can support the research starting from sample preparation, of course, the microscopy part, but also afterwards, the data processing and the data handling. And then we have the fourth division, and it's medical and research part. This is more a prototype right now. It's called the HistoFlow, and it's an AI-powered label-free pathology device, which should be used in the field, in the medical field, more for label-free pathology. I will briefly give an overview about that later. So today, I would like to talk about multiphoton microscopy. And here's an overview of our MPX microscope. The microscope, as I mentioned before, the MPX series is fully integrated, which means that we have the laser, which is already built in the microscope. So you have the controller unit, which you can see under the desk. It contains the light sources, the laser, and also the light source for the wide field technique. And then you have the scan head on top of the table. And then you only need an additional display to start imaging. So the whole design and the whole concept of this microscope is really to address also non-experts. You can build it up by yourself. You don't need a technician to help you for that. It's very flexible and you have a high mobility with this microscope, which means you can build it up anywhere. You don't. The microscope does not require a lot of space. Usually it's built up on a 60 × 60 breadboard, and you don't necessarily need an optical table. Of course, it's beneficial, but you don't necessarily need this. You have a very large working distance, so you can mount any kind of sample underneath the scan head. And you can use the scan head in different configurations, like an upright configuration, inverted configuration, or in different angles, depending on your experiment. This is how the microscope looks like in a real lab, what you can see on the upper row, and also in a more like workshop and environment on the bottom row. You can see we had this workshop, and also we do live demos and on exhibitions. Of course, these are not the perfect environments for the microscope, but it's very nice for us to see that the microscope is also performing in these very bright rooms with a lot of noise, with a lot of vibrations. And on the bottom row, on your right-hand side, you can see the microscope and how it can be moved from one department to another. For example, within university, the controller comes on wheels, so you can just rebuild or disassemble the microscope, bring it to a new place and build it up. And this does not take much time. Today it's about multiphoton microscopy. And as I mentioned before, we have a multimodal microscope, which means we do not only have one modality inside. We have 2-photon microscopy. We are working on 3-photon microscopy in the future. We have higher harmonics microscopy, like. Oh, sorry. Okay. I heard something in the back. I don't think that was a question. So we have higher harmonics microscopy, like SHG and THG. We have the standard wide field epifluorescence microscopy, and you can have additional film microscopy, so fluorescence lifetime microscopy as well. We already heard a lot today about 3-photon microscopy, so I go over the theory very quickly, but maybe for everyone who just joined. So what is the difference between 1-photon and 2-photon, 3-photon microscopy? For 1-photon microscopy, you use a laser in the visible range to excite the fluorophore. And for 2- and 3-photon, you use a laser in the infrared range. And instead of 1-photon exciting a fluorophore, you need 2 or 3 photons hitting simultaneously a fluorophore to detect emission light. This makes this technique intrinsic confocal. What does it mean for the sample? Here you can see a direct comparison between wide field microscopy and multiphoton microscopy. It's a volume scan of a sunflower pollen. It's a sample that is actually not very large, but it's a very nice sample to see the differences. For the wide field microscopy, what you can see on the top, you have a very large focal volume and you only see the blurry light, or only a blurry sample actually when you do the volume scan. To make nice images, like you can see on your right hand side, where you can see the whole slide image of this mouse head. You have to physically slide your sample. So you have to make your sample very thin to get only the light from the focal plane. For the multiphoton microscopy, you have a very small focal volume, and that is why you don't need to physically slide your sample. You can optical slide through the sample, and that allows you to make very nice volume scans, like you can see on the bottom on your right hand side. It's a volume scan of a zebrafish larva. Okay, so what kind of samples can we image with this microscope or with this microscope techniques? So the idea here is a microscope so one size fits all, which means you have one microscope and you can image a broad range of different samples. Of course, multiphoton microscopy is mostly suited for 3D samples. So we start from the microscale with spheroids and organoids, but you go up to the millimeter scale. If you look at, for example, tumor models, tissue engineered constructs, whole slide images, whole organs, or even in vivo mouse imaging. You have a very high flexibility on the sample side because you have this very large working distance that allows you to image a lot of different kinds of samples. You can do live animal imaging, which you can see on your left-hand side. In this case, it was calcium imaging in the mouse brain. You can do whole organ imaging. All of the examples here are in the upright position or upright configuration, but of course you can have your lab dish and then you just invert the microscope, and you still can look at your standard objective slides, of course. Also from the field of view, you're very flexible. You can very easily do whole slide imaging or mosaic scanning. You can have a look on just one field of view, like with a snapshot, and you can go down to the single cell level just by one click. And the kind of sample is also very flexible. So you can still use your standard staining protocols like immunohistochemistry or cytochemistry. But the nice thing, or one big advantage of multiphoton microscopy, is that you have a lot of label-free biomarkers that you can look at, especially collagen. I will review about this a little bit later in more detail. So you can look at fluorophores, you can look at label-free biomarkers, you can use standard FFPE samples, you can use cryosections, or you can use 3D tissue. So you're not limited on the sample side here. So here's an overview of a few examples and applications field that we already worked in, starting from clinical and pharmacological research to neuroscience, in vivo imaging, also tissue engineering, and everything with cell culture, so 3D cell culture, spheroids, organoids, and so on. And in the next part of this talk, I will give a few examples to give you an impression what samples you can image and a few collaborations we already had. So again, we start from the micro to the macro scale, and on the micro scale, 3D cell culture imaging is very important. And you can see on your left-hand side a time lapse of a spheroid, of a living spheroid. And the nice thing here is with multiphoton microscopy, you're not limited to the surface only of a sample. In this case, we look for a imaging plane in the middle of the spheroid, and then we started the time lapse. So you can see what's going on inside of your 3D cell culture. In the middle row, you see animation of a volume scan and a maximum intensity projection. So you can do a volume scan and then have a maximum intensity projection to have all the information on one glance. And on your right-hand side, we call it 2.5 D cell culture. This was a micro patterning of the glass surface. So by an ablation of the surface by laser to make adhesive and non-adhesive parts. And yeah, we wrote in our company logo here. A little bit more advanced spheroids are if you use 3D printed spheroids. This was a cooperation with Fluicell. They do 3D printing of cells without a scaffold. And again, here, it's very important to, if you use 3D cell culture, that you also have 3D techniques to image your samples, right? Because if it's getting more and more complex, if you think about organoids, and you have different cell types, for example, you have very large spheroids, then you really need to know what's inside your spheroids and not only what's on the surface, right? If you go one step bigger, like these tumor models here, these tumor models, they grow ex vivo very large because they are grown on a CAM membrane and they start to build their own vascularization system. And in this case, it's a glioblastoma cell line, which grow very, very dense. And if you have these kind of samples that are not only very large, but also very dense and highly scattering, on the left-hand side, you can see the imaging, just the sample as it was. And we are limited to a depth from 180 micrometer, which is not very deep compared to the size of the sample, right? In this case, you can apply additional post-processing on the sample side, for example, optical clearing to make the sample more transparent. And in this case, after that, we applied the clearing, we were able to image the whole sample, which was 1.3 millimeter in diameter, roughly. So clearing is a very nice tool if you have very large samples and highly scattering samples to improve your imaging depth. Then multiphoton imaging is also very interesting and important in the field of neuroscience. In this case, the university were interested in neuronal tracking. You can see a resected Drosophila brain And what they usually did is that they resected this brain and then they sliced the brain and imaged the slices with confocal microscopy. But if you're really interested in a 3D surrounding, and in this case, neuronal tracking, so they wanted to see the neurons in their native 3D environment, then you need other 3D techniques. And especially they were interested in these antenna neurons, which you can see at the beginning of the volume scan in red in the middle. I did a close-up of the interesting area. And on the right, you can see the individual channels in cyan and in red. There are different neurons populations, and in green, the SHG signal, and in blue, the cell nuclei. And also in the field of neuroscience, in vivo imaging needs 3D microscopy techniques like 2-photon and 3-photon. This is an example of in vivo imaging in the mouse brain to look at the neuron activity. It is a GCamp reporter. And so again, you have the very large working distance under the microscope, which allows you to put also your own stage for your own animal experiments underneath the microscope. And then you can do very easy live animal imaging. Also, if you use the tilting function, for example, so if you need to look at on the brain, for example, in different angles, then you can just tilt the scan head and you can keep the mouse on its upright position. So the mouse feels relaxed. Then, of course, you can do a whole organ imaging, like a whole mouse brain. Then it's more like a snapshot because the brain is fixed, of course, and then cleared. But in this case, the clearing was very nice. So the limiting factor here of penetration depth was really the working distance of the objective, which was 4 millimeters, and we stopped at 3.5 millimeters for the sample. But so this allows you to do very, very large volume scans. We already heard today about zebrafish, and zebrafish are very nice samples. So zebrafish, especially in the larval state, they are kind of transparent, which making them a very useful animal model to do in vivo imaging, also because they are developing very fast and they are used to room temperature. So not like, say, a culture that you need an additional incubation. You can easily image them at room temperature. In the first video, you can see a time lapse over hours of the zebrafish eye. Then in the middle, you can see a volume scan of the zebrafish tail, where you can nicely see the collagen or the muscles in the tail in blue, which are image label free, and then the cell-cell connections in green and some neurons in red. And if you do a very fast scanning, you can also look at very fast movements like the zebrafish heartbeat, which you can see on your right-hand side. Tissue engineering is also a very interesting field for 3D microscopy or multiphoton microscopy, because first you have very large 3D samples you need to image, and also with tissue engineering you can define the growing area of the cells, and with this you also can define a little bit the optical properties of your sample. We have three examples here. The first one is an artificial lung tissue. So this was, so they took a native lung tissue, made a model out of it, and then 3D printed this lung tissue, seeded it with cells. In the second example, you can see an artificial artery. Here it was very interesting. If the cells grow or align along the artery and also start expressing collagen, which we can see with SHG very nicely. And the third example, it's official bone tissue. So you have a lot of collagen here. And again, collagen is very nice, can be very nice imaged with a second harmonic generation microscopy, which is a label-free imaging technique. If you want to look at very large specimens in 2D, so in this case it is a cryosection, which is very thin, but they're very large. You can do mosaic scanning. Here the group was interested in the delivery of antibodies in the mouse, so they applied a therapeutic antibody to the mouse. After that, they sacrificed the mouse, and then they sliced the whole mouse brain into sections, and we did the imaging. First, the whole mouse brain that you really have an overview, and then you can zoom into different sections. This is a very nice tool if you want to look at the parameter in an environmental condition. I mentioned it before, so label-free biomarkers. Big advantage of multiphoton microscopy is that you can look at label-free biomarkers. Wikipedia says a biomarker is a measurable indicator of some biological state or condition. I think that is a very nice description about biomarkers. And of course, there are different properties of biomarkers. So there are optical properties, like the fluorescence of proteins or the absorption, for example, like the absorption of DNA you can use as a biomarker. You have molecular properties like the metabolic state, which you can look at with fluorescence lifetime microscopy or also Raman microscopy. You have mechanical properties like the stiffness of a cell or the stiffness of a tissue. And then more on the microscale, you have morphological properties like the shape of the cell nucleus. It's very important, especially for pathologists. And on the macro scale, the structural properties like, for example, the wall thickness of an epithelium, which can tell you, for example, something about the inflammation state. Where do you need biomarkers? So in the medicine, you can use biomarkers for predictive diagnostic or prognostic indication. And the multiphoton microscopy right now is mostly used in the research field. So biomarkers in the research field are mostly used for contact-free and non-destructive imaging. So this is a nice example about biomarkers where you can use both techniques. We call it wide field guided multiphoton imaging. What you can see here is an FFPE sample of melanoma, a human melanoma. And on the top row, you can see the tissue section image label free. And on the bottom row, you can see the physical H&E staining as a ground truth, because H&E, of course, is the gold standard in the field of pathology to make diagnostic on a sample. If you look at the top row at the label-free imaging, what we are doing is we use the wide field technique, which is a snapshot technique to make a very fast overview scan. And then we decide on different region of interest. And in green, you can see the healthy tissue or areas in the healthy tissue. In blue, you can see the healthy and the lesion. And in red, you can see only the lesion tissue. And we clearly can see differences here. So we can tell that the SHG signal probably originates from the collagen, which you can see in blue. And then you have the autofluorescence in green and in yellow. And the nice thing about the field of pathology or in this area is that you just need a yes or no answer, right? So if you imagine now the FFPE sample to be a cryosection within an operation, then you can just make a very fast section and the pathologist can make a very fast diagnostics because you only need to detect the tumor border here. So you can use the biomarkers, which are not specific here, of course, but you can use biomarkers like the autofluorescence to distinguish between healthy tissue and tumor tissue. Especially collagen is a very nice biomarker when you use multiphoton microscopy, because from fibrous collagen, you get a very bright signal, and you can look at collagen in different kind of samples. You have collagen in 2D tissue, so that you can see on your left-hand side. It's an FFPE section of a column, and you can see the collagen in blue. You can look at the collagen in 3D tissue. On the upper 2 images, you see the collagen in blue from a swine kidney. We just took from the butcher a swine kidney and we imaged this, and you can very nicely see the collagen and also the autofluorescence, which again is not very specific. And also cryosection, like a mouse colon. It's a 200 micrometer mouse colon cryosection, and you can image a collagen shown in blue here, and the red is we applied the staining for the cell nuclei in red, so this is not label free, but the collagen in blue you can see very nicely. And I mentioned it before, collagen scaffolds, because collagen is a very important scaffold in the field of tissue engineering, and to image your scaffold label free tells you a lot of your experiment. It can be very, very useful. But you not only can image the collagen, you also can image muscle. And this is a very nice sample, because here you can see both. You can see the collagen from the tendon, and you can see the muscle tissue. If you look at the overview image in the middle, you can see the very bright tendon running through the image. And if you look on the close-up on your left-hand side, then you can really distinguish between the collagen in the tendon and the sarcomeres or the C-bands in the muscle tissue. And if you do a plot over the C-bands, then you really can distinguish between the single bands very nicely. So I would like to I would like to give an announcement. So we do a hands-on demo tour. The first demo tour will be in Zurich on May 9th to May 17th. First at the ETH in Zurich, and then the second one at the Campus Irche. So if you're interested and you're in this area, please just contact us. You can bring your own samples and you can test the microscope there. And here are a few references. We were very happy that we did the second place from the Willy Analytical Science Award in 2023. And we also just published an article about turnkey multiphoton microscopy for 3D samples at the Willy Scientific Journal. So if you're interested in the examples I just showed in more detail, please check out our references. And of course, I would like to thank all the collaborators we're working with for all the nice samples. And with this, I would like to come to the end of my talk. Thank you very much. And please feel free to ask any questions.